Okay, so our today's topic for discussion is about glycogen. So in the previous class, we have learned about glycolysis and we have seen that how the glucose gets broken. Now in today's session, we will be learning about the glycogen. So what do you know about glycogen? How to define it? What type of carbohydrate is glycogen?
Ma'am, is a storing carbohydrate.
Glycogen is storage form of glucose.
Correct. So which means that this is a polymeric form of glucose. So glycogen is the polymeric form of glucose, which means that the glucose can be converted to glycogen by a polymerization reaction or by a synthesis process. So the process of glycogenesis is the anabolic method in which the glucose they combine together and they give rise to the polymer that is the glycogen. Simultaneously, it may also happen that the glycogen will start to break. And when the glycogen break, then what do you get? You get back the glucose. So in that way, in our body, the amount of glucose and the amount of glycogen both are maintained, right? Now, just to proceed with these two processes, let us find out the most important molecule. If you remember the last class, that the most important molecule in this carbohydrate catabolism or anabolism is it is glucose 6 phosphate. You remember that the second step of glycolysis where glucose gets
Yes, ma'am.
converted into glucose 6 phosphate. And I told that this glucose 6 phosphate can actually relate with different processes. Like we know that the glycogen is a polymer, right? Is a polymer of glucose. And if you remember the last to last class where we have learned about the polymerization process, so we have learned that in glycogen, what type of linkages you get to see? What linkages you get to see in glycogen? If you remember, tell me. In glycogen, what type of linkage you will get? Linear or branched?
Branched.
Branched.
Branched, which means that
Branch.
there will be both C1C4 linkage and then the C1C4 linkage will again combine with the help of the C1C6 bond, which means that the branching is possible here because the sixth carbon is involved here and the fourth carbon is also getting involved here. So we will find that in order to form the bond, it is necessary that the glucose which we are having, it must be converted into glucose 1 phosphate. So the glycogen is produced from glucose. How? By generating glucose 6 phosphate. And this glucose 6 phosphate is converted into the glycogen. Now, when the glycogen is broken, then what is happening? Then the glycogen first give rise to glucose 1 phosphate. Why? Because you remember we have learned that in the chain there was C1 and C6 which were present. So when you break the glycogen, you get glucose 1 phosphate. And then this glucose 1 phosphate is converted back into glucose 6 phosphate. Which means if you are learning about the glycogen synthesis, then the process will get diverted from glucose 6 phosphate. And if you're learning about glycogenolysis, then the method will be starting from glycogen. It will lead to glucose 1 phosphate and then it will convert into glucose 6 phosphate. So from here, we need to identify that these two methods, they are dependent on these two molecules, that is glucose 1 phosphate and glucose 6 phosphate. Fine. Now, other than these two molecules, there are two major enzymes which are going to regulate the processes. So these enzymes actually help or they decide that whether the glycogen will be formed or whether the glycogen will be broken. Now, what are these two enzymes? Like here you see in this particular stage, what we can find that the glycogen is breaking up, right? So how the glycogen is breaking up? The glycogen is breaking up by the activity of the enzyme called glycogen phosphorylase. So this we need to remember that if you are synthesizing glycogen, right? If you are synthesizing glycogen, then the enzyme that you will be requiring here is glycogen synthase. यानी glycogen अगर हमें prepare करवाना है, जो enzyme हमें चाहिए होगा, that will be glycogen synthase. But then, which we call as GS. In short, you can write it as GS. But then, if you are doing the reverse process, yani ki if you are breaking the glycogen to give rise to glucose, then the enzyme that you will be requiring here is glycogen phosphorylase, which means that these two enzymes are just the opposite. You see, glycogen synthase is synthesizing glycogen, glycogen phosphorylase is breaking the glycogen. Correct? And these two enzymes, they are actually inhibiting each other, which means that if the glycogen phosphorylase is active, if glycogen phosphorylase is active, then glycogen synthase will become inactive. Simultaneously, if glycogen synthase is active, then the glycogen phosphorylase will become inactive. So, the glycogen synthesis will occur when glycogen synthase is working and phosphorylase is resting, is inactive. And again, when this glycogen phosphorylase gets activated, then the glycogen will start to break and it will give rise to glucose. Right? So till now what we have learned, that the two processes, that is your glycogenesis and glycogenolysis, these two processes depend on the two major molecules, glucose 1-phosphate and glucose 6-phosphate, and the two controlling enzymes, that is glycogen synthase and glycogen phosphorylase. Do we understand this much? The enzymes
Ma'am,
and the molecule. Yes, Shohini?
ma'am, what is it is shown here कि glycogen N-1. What is it?
It is a polymer, right? Polymer means that there are multiple glucose which are present in glycogen. So let us consider there are n number of glucose which are present in glycogen. Now, when you are breaking the glycogen, means what? That from a long chain of glycogen, you are taking only one glucose. So if you are taking one glucose, then what is left? You are left with glycogen n-1. n number of glycogens थे, अब उसमें से तुमने एक ही ग्लूकोज को जो है यहाँ से रिमूव किया है. तो अब तो जो बच गया वो
Yes,
क्या
ma'am.
बोलते हैं? Yes, we will call it as glycogen n-1. Just tell me that do we understand the two enzymes? The two
Yes, ma'am.
enzymes which
Yes,
are
ma'am.
Yes, ma'am.
Yes, ma'am.
now the question is that when these processes are going to happen. Like tell me that say for example a person is starving, right? Or a person is doing excessive exercise. So what do you think that what is the process that must occur in the body, glycogenesis or glycogenolysis? Glycogen synthesis will happen or glycogen breakdown will happen if a person is starving or doing exercise.
Glycogen breakdown.
Glycogen breakdown.
Glycogen breakdown.
Ma'am, breakdown.
Simultaneously, correct. Simultaneously, if a person is in a well-fed state, which means a person is eating a lot amount of food, then definitely the glycogen will be formed, right? So we need to understand that these two processes, they are actually reciprocal processes, which means that these two processes, they are just the opposite and they occur in two different versions of our body. So one such phase will be a well-fed state. Well-fed state means that in our body, there is enough food, we are eating a lot, and there is enough amount of glucose. So when there is excess glucose, then this excess glucose will be converted into glucose 6-phosphate. You remember for glucose synthesis, the direct molecule that you need is the glucose 6-phosphate. So when you eat a lot amount of glucose, it gets converted into glucose 6-phosphate, which ultimately give rise to the glycogen. Simultaneously, you will see that this glucose 6-phosphate, it is again generated back when the glycogen is getting broken. So when you start the process of breakdown of glycogen, so you ultimately get this glucose 6-phosphate back, and this glucose 6-phosphate can then be utilized by the body. Like say for example, the person was starving, right? So the glycogen got broken, the glucose was released, the glucose 1-phosphate was converted into glucose 6-phosphate, and then this glucose 6-phosphate enter into glycolysis. So as because the person was starving, so this glycolysis will give energy to the person. or the pentose phosphate pathway, which means this is a pathway that we will learn in the next class. So you will see that this is a pathway where excess amount of energy is needed. Say, for example, in one part of your body, excess amount of energy is needed. So who will give it? Pentose phosphate pathway will give it. So when excess amount of energy is needed, then also the glycogen is broken, the glucose is released, and then it goes to the pentose phosphate pathway. Other than that, the conversion of the free glucose for transport to other organ. Like it may happen that suddenly there is a fall of glucose in brain, right? The brain is starving from glucose. So then also the glycogen breaks and the glucose which is released, it will go with the blood flow and it will reach the brain. So this is the function of glucose in our body, which is generated by the breakdown of glycogen. Fine. So from
So,
here,
ma'am,
we yes, Devamita.
from glucose 6 phosphate is from glucose we are getting glucose 6 phosphate, and
Correct.
again from glycogen also we are getting glucose 6 phosphate.
Yes. See, it is a linkage, like I said, right? This is a link. Like, say, for example, if you are going from a ground floor to the first floor, then you are using a stair, right, to go up.
Yes,
Simultaneously, अगर तुम सीढ़ी से नीचे आओगे, then you are using the same staircase while you come
yes,
down. In the same way, you have a glucose and you have a glycogen, and the intermediate is glucose 6 phosphate, right?
okay,
तो तुम ग्लूकोज से ग्लाइकोजन बनाओ, तुम्हें ग्लूकोज 6 फॉस्फेट होके ही जाना है. तुम ग्लाइकोजन से ब्रेक होके तुम ग्लूकोज में आओ, तुम्हें ग्लूकोज 6 फॉस्फेट होके ही आना है, राइट? सो दिस इज अ कॉमन मॉलिक्यूल.
okay.
So yes, so we need to remember that there are two major enzymes which we need to learn, that is glycogen synthase and glycogen phosphorylase. So let us see the function of this enzyme first, that is glycogen phosphorylase. Now this
Ma'am, ma'am, ma'am,
yes,
can you please repeat once what happens during starvation?
okay. During starvation, there is less glucose available, right? And it
Yes,
may
ma'am.
happen that there is glucose deprivation in brain. The brain is lacking the glucose. So at that time, the body has to give rise to glucose. So what it does, it breaks down the glycogen. Like say, for example, we know that glycogen is there in our liver. Now the liver is experiencing that there is less amount of glucose available. So this liver will start breaking the glycogen. It will release glucose 1 phosphate. It will convert glucose 1 phosphate into glucose 6 phosphate, and then glucose 6 phosphate will finally give rise to glucose, right? So that is how they are actually related to each other. So whenever there is a fall in the glucose concentration, the glycogen molecules are broken so that glucose can be released during starvation.
Okay, ma'am. Thank you.
Yes. So this is your enzyme, that is glycogen phosphorylase. Now what we have learned, that when this enzyme is going to work, when we have to break the glycogen. So glycogen को तोड़ना है, then we need glycogen phosphorylase. Fine. Now if you see this enzyme very minutely, why we will see that? So first of all, let us find out that what is the structure. So you see that this enzyme has a dimeric structure. Dimeric means there are two units, and in this two unit, there is a catalytic site like every enzyme is having, and there is a allosteric binding site and there is a phosphorylation site. यानी कि ये जो एंजाइम है, ये एंजाइम अपना कैटालाइसिस तो करता ही है, यानी कि उसका जो नॉर्मल फंक्शन है कि ग्लाइकोजन को ब्रेक डाउन कराना है, दैट इट विल डू. Other than that, this enzyme has two special properties. What? It can bind with the allosteric site, allosteric binding site. You know that every enzyme has a activity site and it may also have a allosteric site. तो allosteric मतलब inhibition site. जिस साइट पे एंजाइम को बाइंड करके हम एंजाइम को ब्लॉक करा सकते हैं, यानी कि इस एंजाइम को कोई दूसरा मॉलिक्यूल आके ब्लॉक कर सकता है. So this is a speciality of this enzyme, that this enzyme has allosteric site, which means it can be blocked by some other molecule. Now, there is another site. What is it? That is your phosphorylation site. Phosphorylation site means the site where phosphate is added. Correct? So we need to remember that this enzyme is special. Why? Because this enzyme can bind with phosphate and it can also get inhibited by any other molecule. So you will find that this enzyme, as because it can bind with phosphate, so it has two types of variation. यानी कि एक वेरिएशन में हमें दिखेगा that it is phosphorylated, which means that if the enzyme is bound to the phosphate, then I will say that the glucose phosphatase is active enzyme, which means that whenever this enzyme is bound to phosphate, तो अगर इस phosphorylation site पे अगर phosphate bounded है, यानी कि the enzyme is active. Simultaneously, if there is dephosphorylation means removal of phosphate. So if the phosphate is removed from the site, then this enzyme will become less active or inactive. Just understand this कि ये जो एंजाइम है ग्लाइकोजन फॉस्फोराइलेज, ये कैसे एक्टिव होता है? जब इसके साथ फॉस्फेट बाइंड होता है देन इट बिकम्स एक्टिव. जब इससे हम लोग फॉस्फेट हटा देते हैं सो इट बिकम्स इनएक्टिव. दिस इज द प्रॉपर्टी ऑफ दिस एंजाइम. Tell me, do we understand this?
Yes, ma'am.
Yes, ma'am.
Okay,
Yes, ma'am.
correct. Now, let us see that how this enzyme is going to take part. Yes. So we have learned that there are two major groups of enzymes, right? क्या क्या पढ़ा था हमने? Glucose, glycogen synthase and your glycogen phosphorylase. तो glycogen phosphorylase क्या होता है? इसमें क्या क्या parts है? ये तो हमने देख लिया. Now we will see the next enzyme, that is your glycogen synthase. Now, what is the purpose of this glycogen synthase enzyme? To synthesize glycogen, है ना? Glycogen बनाना है. And how this glycogen will be synthesized? By combining the glucose. यानी कि when there is lot amount of glucose in the body, so all the glucose molecules, they will be arranged one after the other and a long chain of glycogen will be formed. So who will do this? This will be done by the enzyme glycogen synthase. Now we will just see that how this enzyme does this. So yes, so we have learned that the process will begin from glucose. Body में बहुत ज़्यादा glucose है. So this glucose will get converted into glucose 6-phosphate. By which enzyme? Glucokinase. If you remember the last class, can you tell me that where will you find this glucokinase? Where will you find
Ma'am,
glucokinase?
glycolysis.
Glucokinase is present in all body parts.
Ma'am,
Liver.
Ma'am, liver.
Liver and pancreas.
and pancreas.
Liver and pancreas. Yes, liver and pancreas, mostly it is found in the liver. So here you see here this entire process that is shown, it is actually occurring where? This process is actually occurring in the liver. Correct. So what happened? That the glucose which was there in the liver, in presence of glucokinase, it broke into glucose 6-phosphate. Now, what we know कि हमें अगर पॉलीमराइजेशन कराना है तो हमें कौन से कार्बन पे फॉस्फेट चाहिए? फर्स्ट कार्बन पे फॉस्फेट चाहिए. यानी कि ग्लूकोज के जो 6th कार्बन पे जो फॉस्फेट है, इट मस्ट बी रिमूव फ्रॉम द 6th कार्बन एंड इट मस्ट बी अटैच टू द फर्स्ट कार्बन. क्योंकि जब ग्लूकोज के फर्स्ट कार्बन पे फॉस्फेट आएगा देन ओनली इट कैन स्टार्ट फॉर्मिंग ऑफ द polymeric chain of glycogen. So there will be a change in the position of the phosphate. How? By the phosphoglucomutase. Mutase means change. Changing what? The position of phosphate in glucose. So we call it as phosphoglucomutase. So you will see that this enzyme is going to change the phosphate from 6th carbon to 1st carbon. So you've got glucose 1-phosphate. So you are ready to begin this process of glycogen synthesis. Now, for this reaction, we need the enzyme glycogen synthase, right? Now, just remember that glycogen synthase is an enzyme which has different subunit. यानी ग्लाइकोजन सिंथेज जो होता है ना, वो अकेला काम नहीं करता, वो अपने साथ और बहुत सारे मॉलिक्यूल्स और बहुत सारे एंजाइम्स के साथ काम करता है. So what happened that when this glucose 1-phosphate is generated, first of all, this glucose 1-phosphate will bind with a UDP molecule. यानी हमारे पास ग्लूकोज था, ग्लूकोज को हमने ग्लूकोज 1-phosphate बना दिया, but then the glucose 1-phosphate directly cannot bind with one another. So in order to bind with one another, it need a molecule called UDP. Can anyone tell me what is this UDP? Good.
Ma'am, a uracil diphosphate.
Okay, like we know that there is ATP, there is ADP, right? In the same way, there is UDP. So this is another energy currency molecule. So this UDP, it will bind with the glucose. तो glucose अकेला जो है ये process conduct नहीं कराएगा. Who will bind with the glucose? The UDP molecule will bind. So you see that this step shows that how the UDP got bound with the glucose. Now after this, we have one glucose which is bound to UDP. यानी कि हमारा जो प्रोसेस है उसको शुरू करने के लिए हमें क्या चाहिए, ये हमें ये समझना है कि जब ग्लाइकोजन बनता है तो फर्स्ट एक ऐसा ग्लूकोज होगा जो यूडीपी के साथ बाइंड करेगा, एंड देन दिस ग्लूकोज विल बाइंड with a big molecule called glycogenin. What is glycogenin? Glycogenin is a primer. Primer मतलब जैसे सोच लो एक स्मॉल स्ट्रेच है, एक स्मॉल एक यूनिट है जिसके साथ एक के बाद एक हम ग्लूकोज ऐड करते जाएंगे. यू विल कीप ऑन एडिंग ग्लूकोज टू दिस ग्लाइकोजेन एंड अल्टीमेटली यू गेट द ग्लाइकोजेन. तो ये जो एडिशन है, यू आर एडिंग समथिंग, राइट? सो यू विल एड इट टू अ मॉलिक्यूल और टू अ स्ट्रक्चर. किसी भी चीज को जब हम ऐड करते हैं तो उसको ऐड करने के लिए, उसको होल्ड करने के लिए, उसको जोड़ने के लिए हमें कुछ एक बेस चाहिए होता है जिसके ऊपर तुम उस मॉलिक्यूल को ऐड करोगे. So for this glycogen, what is your that primer? What is your that base? That is your glycogenin. यानी पहले ग्लाइकोजेनिन मॉलिक्यूल आएगा, वो एक ग्लूकोज के साथ बाइंड करेगा एंड इट विल स्टार्ट द प्रोसेस ऑफ एडिंग मोर एंड मोर ग्लूकोज टू इट. सो व्हाट वी हैव लर्न that in glycogen synthesis, जरा starting से देख लेते हैं. In glycogen synthesis, all the glucose which is present, it gets converted into glucose 1-phosphate, which binds with the energy molecule called UDP, and they bind to a primer called glycogenin, and then they are ready to initiate this process. Do we understand this much?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes. So
Yes,
you
ma'am.
see, ultimately what you get here is called your glycogen primer. So your primer is now ready. Now, to this primer, you will add more and more glucose. So you see that you are adding how many glucose here? You are adding 13 glucose. And for every glucose, you are also giving a UDP. So whatever glucose will come, it will come with the help of a UDP. So once your primer is ready, then one after the other, the glucose will keep on coming and they will form a long chain. तो हमें देखो क्या मिल गया, ग्लूकोज का एक लीनियर चेन मिल गया. You got the linear chain, right? But the problem is that we have learned that in glycogen there is branching. पर हमें तो यहाँ पे linear chain मिला. There is no branching yet, which means that after a linear chain is created, the next step will be to introduce branching. यानी कि हमें अभी क्या चाहिए होगा? We will be requiring the branching enzyme, the enzyme which will introduce branching. So you see that to this linear chain, what is happening? To this linear chain, a new molecule is adding up, which is called as your glycosyl transferase. यानी कि इस एंजाइम का काम क्या है? ट्रांसफर करना. किसको ट्रांसफर करना? ग्लूकोज मॉलिक्यूल्स को ट्रांसफर करना. So how the glucose molecules are getting transferred? You see, यहाँ पे कितने ग्लूकोज थे हमारे पास? We were having 16, 1 to 16 number of glucose were added. Now, this enzyme, what it will do? It will break a stretch of glucose from here and it will bind it here. यानी कि एक लंबा चेन था, उसमें लास्ट के यूनिट्स को तोड़ के हमने एक ब्रांच जनरेट किया. So this enzyme here, that is your glucosyl transferase, is a branching enzyme which can generate the branching in a linear chain of glucose. Right? So do we understand that first what happened? A single glucose bind to the glycogenin molecule, a primer is created, then after which long glucose chain is added up. So this linear chain is produced, from where the linear chain again undergoes a branching mechanism and a branch unit is created. Right? Is this clear to you?
Yes, ma'am.
Yes, ma'am.
Right. So you see that in this process, first of all, a linear chain was formed and then branching was done, right? After which the same process will be repeated. As because this is polymerization, right? So polymerization means what? The same step is repeated again and again to give rise to a polymer. So these are the two steps which are going to happen again and again. What is it? A long glycogen synthase enzyme will elongate the chain, that is a linear chain, and in the next step, again branching will happen. So this process will continue unless you get a big molecule of glycogen. So in glycogen synthesis, there are actually three major steps which are happening. What is that? In the first step, the glycogenin is binding up with the UDP glucose. In the second step, The glycogen synthase is creating a long chain, a long linear chain. In the third step, the glucosyl transferase enzyme is creating branching. And then the same process is getting repeated unless and until you get a big glycogen. So in this process, we need to remember that there are two types of enzymes which are involved. What is it? One is elongation enzyme, that is your glycogen synthase. Another is branching enzyme, that is your uh, glucosyl transferase. Just check. Do you understand this process? Whatever we have learned till now.
Excuse me, ma'am. Ma'am,
Yes.
what is? Can you explain the portion of UDP and glycogen? Glycogen is an enzyme that is helping UDP to bound to the glucose.
Okay. Basically, glycogenin is a self-phosphorylating protein. It is a
Okay.
self-phosphorylating protein, right? And it is said that this works with the glycogen synthase enzyme. Like I told you, the glycogen synthase works with many enzymes. So you see that this is one glycogen synthase which has different subunit. So one subunit is glycogen initiator synthase. The other subunit is glycogenin. The third subunit is glycosyl transferase. So these are the different units of the same enzyme which work together. So basically, if you ask that what is glycogenin, so this is a primer which is actually a self-phosphorylating protein. You
Okay, ma'am.
understand that?
Yes, ma'am.
Okay, right. So this is your uh, glycogen synthesis process. So see, what we have learned here, we have, just to sum up, we have learned that the glycogen, in order to get created, it requires the UDP glucose, the most important thing. And it also requires the molecule that is glycogenin. And in the process, there are two major steps that we need to remember. First, there will be elongation. So a long chain will be created. And then there is the branching enzyme, which will generate the new bond to form a branch. So that is how the big glycogen molecule is formed, right? Now, we will quickly see the regulation of this process. Now, this process was about formation of glycogen. So we call it as the glycogen synthesis or glycogenesis. So we need to remember that in this process, there are activators and there are inhibitors. So what is the activator? Insulin is the activator. What we know that what is the purpose of insulin? To lower down the blood glucose, है ना? यानी कि अगर ब्लड में अगर फ्री ग्लूकोज है तो उस फ्री ग्लूकोज को ब्लड से हटाना है, which means that whatever free glucose is there in the blood, it should be combined together to form a glycogen. क्योंकि देखो जब ग्लाइकोजन बन रहा है तो क्या हो रहा है? The free glucose which is suspended, it is combining up together to give rise to a bulky molecule of glycogen. यानी कि at the time of glycogen formation, the blood glucose level, it is increasing or it is decreasing when glycogen is forming up? What do you think?
Decreasing.
It's
Decreasing.
decreasing. Correct. It's
Decreasing.
decreasing. Yes, it decreases. And we know that what is the purpose of insulin? To decrease the blood glucose level. Which means that if there is insulin in the blood, so this insulin will help in conversion of the free glucose available and convert it into glycogen. Like that, the free glucose in the blood will get reduced. Which means that insulin will promote glycogen formation or glycogen synthesis. And that is how it reduces the blood glucose level. Simultaneously, there is another hormone which will do the reverse. What is it? That is your glucagon. So what you know, glucagon, what is the purpose of glucagon? Function of glucagon, what you know?
Increase the glucose in blood.
Increase the blood glucose, which means that now you want to increase the blood glucose. So how to increase the blood glucose? By breaking glycogen. If you break glycogen, then only your blood glucose levels will be high, which means that you want to break the glycogen, you don't want to make the glycogen, which means that in presence of glucagon, there will be no glycogen synthesis, there will be glycogen breakdown. So we can say that glucagon is going to inhibit the process of glycogenesis, whereas the insulin is going to activate the process of glycogenesis. Correct? Other than that, there is another important hormone which will play a very vital role, and that is epinephrine. Epinephrine, do we know what is the purpose of epinephrine? Anyone? Or when it is released by our body? Epinephrine,
Ma'am, it is a stress hormone, if I'm not
stress
wrong.
hormone. Correct. Stress hormone. Like say for example, you are
Ma'am, I
feeling.
got,
Huh,
excuse me, ma'am, I got disconnected. So, could you just
I will share
repeat
the
the last
link.
part?
I'll share the link. You can check. Yes. Okay. So the epinephrine, like we were talking about, it is a emergency hormone, right? Which means that, say for example, you have to run, you are in danger, and you have to run. So while you are running, you need more amount of energy, correct? So in order to get more amount of energy, you need more amount of glucose. And how do you get the glucose? By breaking down the glycogen, which means that the epinephrine, what it has to do, it will help in glycogen formation or it will help in glycogen breakdown. It must give
Glycogen
you,
breakdown.
yes. So it will give you glucose, which means it is going to promote glycogen breakdown. So glycogen breakdown will be promoted simultaneously. Glycogen synthesis will be inhibited or reduced. So you see, it is said that this epinephrine, its action site is muscles. Like what happened that when there is a person who is doing exercise, so the person is requiring more amount of energy. So at that time, the epinephrine, it will reduce the glycogen synthesis and it will stimulate the glycogen breakdown so that the energy can be given to the muscle cells. Fine. So we need to remember that there are three hormones which are regulating the process. Insulin is promoting, glucagon and epinephrine are inhibiting what? They are inhibiting the glycogen synthesis method. Yes, see, do you understand this hormonal regulation?
Ma'am, can you just
Yes,
repeat it
ma'am.
once?
Yes. Just remember one thing, that insulin will promote the process of glycogen synthesis. The purpose of insulin is to reduce the blood glucose. Now, how to reduce the blood glucose? By converting the blood glucose into glycogen, which means that insulin will trigger the process of glycogen synthesis or glycogenesis so that the blood glucose can reduce and glycogen can be formed. Simultaneously, what is the opposite hormone of insulin? That is glucagon. So this glucagon will inhibit the process, it will stop the process. And then the epinephrine, which is also there present in our body, it also makes sure that the process of glycogen synthesis is inhibited. Why? Because what is the purpose of epinephrine? The purpose of epinephrine is to break glycogen. Why? Because when you break the glycogen, you get the energy, right? So from here, just remember one thing, that insulin promotes glycogen synthesis, glucagon and epinephrine inhibit glycogen synthesis.
But both the process cannot occur simultaneously.
Both the process cannot occur simultaneously because you see, when insulin is there, then glucagon concentrations are less in the blood, right? When
Okay,
glucagon is high, then insulin is less.
okay.
Yes. So we have
Got
seen
it.
that what is, yes. So we have seen that what is the action of the enzyme, right? We have seen that what is this insulin and what is this glucagon and what they are going to do. Now here you will see that how they are going to do it. Like we have learned that this insulin will activate and glucagon will stop. It will inhibit. Now the question is that how it is happening. So just to answer this, that how it is happening, do you remember that we have just now learned about this enzyme called glycogen phosphorylase? अभी एक हमने enzyme देखा था ना, glycogen phosphorylase. It was having a
Yes, ma'am.
catalytic site. It was having a,
Allosteric binding site
yes,
Yes,
allosteric
ma'am.
binding site, a phosphorylate site. So I told that it can bind with the phosphate and it can
to be
also remove
active.
the phosphate. Correct. So just remember, just follow this. That this enzyme, अगर glycogen phosphorylase है, यानी वहां पे glycogen breakdown होगा. Correct. And there was another enzyme I said, that is your glycogen synthase. So this glycogen synthase will do just the reverse thing, which means that in presence of phosphate, your glycogen phosphorylase enzyme will become active. And the other enzyme, which is just the reverse, that is your glycogen synthase, in present, it is active without phosphate, which means that if there is no phosphate, then it will remain active. यानी अगर phosphorylation body में हो रहा है, अगर phosphorylation body में हो रहा है, so the phosphate is getting added. Phosphate add होना मतलब कौन सा process अभी होगा? Phosphate add होने से बताओ, which process should happen?
Glycogen breakdown.
Glycogen breakdown. Just follow this. अगर phosphate होता है, so which molecule is getting active? Glycogen phosphorylase is active. If glycogen phosphorylase is active, then your glycogen will break. Simultaneously, what is the reverse process? If there is no phosphate, then your glycogen synthase will become active and the glycogen synthesis will occur. Just understand this much कि अगर phosphate है तो glycogen breakdown होगा, phosphate नहीं है तो glycogen formation होगा. Just understand this much. This much is clear or not?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay,
Yes,
fine.
ma'am.
Yes. So we will quickly see the mode of action, that how it is happening. See, this is the molecule that is your insulin. We will be looking for two different hormones. See how these hormones are doing this process. So the insulin hormone, what it will do, as because it is a hormone, it will go and it will bind to the cell surface receptor. यानी कि body के जो cells हैं, हर cell के membrane पे कुछ ना कुछ receptor होता है. So the hormones, the enzymes, they can go and they can bind there. So insulin being a hormone, it will also go and it will bind to the receptor which is there in the cell membrane, and that is your tyrosine kinase receptor. You don't have to remember the name. Correct? Just remember one thing, that it is going to bind to a receptor whose name is having kinase. Kinase means, you remember we have learned. Kinase means, what was the purpose of kinase? Hexokinase, phosphofructokinase. Kinase means what group of
To
enzymes?
break down.
Ma'am, to
Remove.
Adding phosphate.
Adding phosphate
adding
or to
phosphate.
remove phosphate, correct? Adding phosphate or to remove phosphate. So what happened, that whenever insulin, it binds to this receptor, that is tyrosine kinase receptor, so this receptor will need a phosphate. It will take up a phosphate. And from where it takes up the phosphate? It takes up the phosphate by removing. It takes up the phosphate by removing it from the enzyme. तो जितने भी एंजाइम्स अवेलेबल होंगे सेल्स में, उन सारे सेल्स में से उसने क्या किया? फॉस्फेट को अपटेक कर लिया. कब? जब इंसुलिन आके सेल सरफेस पे बाइंड हुआ. तो जैसे ही सेल सरफेस पे इंसुलिन बाइंड हुआ, दिस kinase receptor enzyme got activated and it started taking up the phosphate. तो जितना भी phosphate था वो हमारे enzyme से हट गया, which means that at this moment the glycogen synthesis will become active क्योंकि phosphate हमारे पास नहीं है, and your glycogen phosphorylase, it will become inactive. So what do you know, that what happens when insulin is there? Which process will take place? Glycogen क्या होगा, बनेगा कि टूटेगा?
Synthesis.
Ma'am, glycogenesis.
Yes, glycogenesis or glycogen synthesis. So you see, so glycogen synthesis occurs because the insulin is helping to remove the phosphate and making this enzyme active. At the very beginning I said कि जब glycogen synthase जब active होता है, तब glycogen phosphorylase inactive हो जाता है. Now do you understand why it happens? क्योंकि glycogen synthase जब active हो रहा है, it is getting activated because phosphate is removed, right? And at the same time, your glycogen phosphorylase, what is happening to them? It is getting deactivated because the phosphate is getting removed. Simultaneously, you see the function of glucagon, just the opposite function it will do. So this glucagon enzyme, hormone, when it binds to the cell surface, then it binds to another receptor. तो जो इंसुलिन था वो टायरोसिन काइनेस के साथ बाइंड हो रहा था, जो ग्लूकागॉन है वो जी कपल्ड के साथ बाइंड होता है. यू डोंट नीड टू रिमेंबर द नेम, जस्ट रिमेंबर वन थिंग, that when it binds to the cell membrane, then it generates the activity of kinase. Activity of kinase yani ki now this kinase will add the phosphate. So as because the phosphate is added up, so this time what is going to happen? Now your glycogen synthase enzyme, it is getting inactivated. Simultaneously, your glycogen phosphorylase enzyme, it is going to get activated because you are adding up the phosphate. Which means that in presence of glucagon, the phosphate is added and the glycogen phosphorylase enzyme is activated, for which your glycogen breakdown will begin. Do you understand this? Just see.
Yes.
Yes, ma'am.
So from here, we need to be very much clear about the functioning of these two enzymes, that is insulin and glucagon. So just see, when the insulin levels in the body are high, at that time, the glycogen synthesis is occurring, which means that when you take the food, insulin level सबसे ज़्यादा high body में कब होता है? जब हम खाना खाते हैं. So when we take the food, that is the actual time when the glycogen is building up in our body. And when the glucagon is high, that is during the starvation period, the glucagon concentration increases in the blood and the glycogen breakdown also increases in the body.
But ma'am, addition of phosphate is occurring in both the cases.
No. Addition of phosphate is occurring with the help of glucagon in the second case. In the first case, the phosphate was removed. You see, phosphate is getting removed. Why? The phosphate is removed from the enzyme. It is going and binding to the receptor. The receptor is taking away the
Okay.
phosphate. So in this case,
Yes.
the phosphate is getting removed. And here the phosphate is getting added.
Okay.
Right? So now do we understand that what exactly happens during starvation and a well-fed situation? How the glycogen synthesis begins and it stops in both the processes?
Yes, mom.
Sorry.
Yes.
Yes. So this is what we have discussed just now, that the insulin which is produced by your pancreas in a well fed state, like I said, कि जब हम खाना खाते हैं तब हमारा well fed state है, right? So at that state there will be a rise in insulin, there will be increase in insulin, there will be an increase in GLUT4. ये भी हमें पता होना चाहिए कि जो इंसुलिन होता है ना, it performs many things. So when insulin is released, first of all it is going to increase the activity of glucose transporter, यानी कि जितना भी ग्लूकोज सेल के बाहर है पहले तो उसको सेल के अंदर लेके आओ. Who will do it? ग्लूकोज ट्रांसपोर्टर विल डू इट. तो ग्लूकोज ट्रांसपोर्टर को एक्टिव किया किसने? इंसुलिन ने. सारा ग्लूकोज कहाँ आ गया? मसल्स के अंदर आ गया. अब मसल्स के अंदर आने के बाद व्हाट दिस इंसुलिन इज गोइंग टू डू? इट विल एक्टिवेट द एंजाइम glycogen synthase so that the glycogen synthesis can take place. So that is how your insulin after you eat will help your muscles to build up the glycogen. Simultaneously, the glucagon, when it is secreted, when your blood glucose level was low, so when you were in the fasting state, at that time the glycogen will start to degrade. Why it will degrade? So that the glucose can be released by your liver and it can get into the body circulation. So only liver cells are rich in glucagon receptors, and therefore respond to this hormone. This is a important part. See, ग्लाइकोजन हमारे बॉडी में दो जगह स्टोर होता है ना? कहाँ कहाँ पे स्टोर होता है बताओ? One is liver, another is muscle, right? Now
Yes,
answer me.
ma'am.
Yes, now answer me that say for example a person is starving, ठीक है? तो उसकी बॉडी में ग्लूकोज अभी कम है. Now at this point the body has to break the glycogen, it has to release the glucose. So out of the liver and the muscle, what do you think that which will release the glucose first? लिवर और मसल में से कौन जल्दी ग्लूकोज देगा बॉडी को?
Liver.
Ma'am, liver.
Why liver? Why liver? क्योंकि लिवर के पास क्या है? ग्लूकागॉन हार्मोन का रिसेप्टर है. ये जो हमने रिसेप्टर अभी देखा कि जो ग्लूकागॉन होता है, ये ग्लूकागॉन कैसे बाइंड कर रहा था? ये जी कबल रिसेप्टर के साथ बाइंड होके इट वाज एडिंग अप द फॉस्फेट एंड देन ओनली योर एंजाइम वाज गेटिंग एक्टिवेटेड, राइट? सो ये जो हमने जो ये प्रोसेस देखा, ये प्रोसेस कौन से सेल्स कर सकते हैं? ओनली लिवर सेल्स कर सकते हैं, बिकॉज योर लिवर सेल्स ओनली हैव दिस रिसेप्टर ऑफ ग्लूकागॉन. योर मसल सेल्स डोंट हैव इट. दैट इज वन रीजन. व्हाट इज द अदर रीजन? कि ग्लाइकोजन ब्रेक डाउन कराने के लिए विच एंजाइम वाज नीडेड इन ऑर्डर टू ब्रेक डाउन द ग्लाइकोजन? बोलो.
Phosphorylase.
Glycogen phosphorylase.
Glycogen phosphorylase. Remember, this glycogen phosphorylase is present in the liver cells only. ये liver cells में ही होता है, muscle के पास नहीं होता है. Which means that again, due to this enzyme, the liver cells can only give rise to free glucose. तो जो ये free glucose का जो concept है, ये only कौन बना के देता है हमेशा? ये हमेशा हमें लिवर ही देता है, मसल्स ये हमें डायरेक्टली नहीं दे पाता है, राइट? So when you are under starvation, at first the liver will start giving you the glucose. After a while, जब हम हमारी बॉडी को जब ये फील होगा कि जो जितना ग्लूकोज लिवर दे रहा है उतने ग्लूकोज से भी नहीं हो रहा है, द बॉडी नीड्स मोर ग्लूकोज, राइट, ड्यूरिंग स्टारवेशन. So then afterwards your muscle will start generating the glucose with the help of the Cori cycle. तो जो Cori cycle है वो start होगा, which is another process of gluconeogenesis, and that is how the glucose comes to your body. So direct glucose is not generated by the muscle, it can be only generated by the liver. Is this clear to all?
Yes, ma'am.
Yes.
Okay. Yes. Now, this is one important fact that we need to know. That what we have seen, that if there is insulin, then what was happening? There was more glycogen synthesis. Correct? यानी कि अगर इंसुलिन ज्यादा है बॉडी में, then there will be more glycogen formation, right? Now tell me one thing, that say for example there is a diabetic patient, ठीक है, उस डायबिटिक पेशेंट को हम जो है प्रोलॉन्ग टाइम से इंसुलिन देते हैं, डेली जो है वो इंसुलिन ले रहा है. सो व्हाट डू यू थिंक that what will happen to the glycogen in the body of that patient? क्या होना चाहिए बताओ? The patient is taking insulin for long. So what is supposed to
Glycogen will be lowered
be lowered? Why?
because
Why?
insulin uh,
Hmm.
because the person is getting insulin from outside, so the amount of insulin will be higher.
The amount of insulin will be higher. So if insulin is higher in the body, then what happens to glycogen?
Breakdown.
Right, a breakdown happens. If
Ma'am,
insulin
is
is
a
higher,
synthesis.
Storage.
synthesis.
Oh, sorry. Synthesis.
So,
Synthesis.
so glycogen synthesis will happen.
Storage.
Storage will happen. Correct. Now, just imagine the situation that there is a diabetic patient who is taking insulin for long, which means that for long time, the glycogen is building up in the glucose, in the liver. यानी कि जो पेशेंट होगा ना उसकी बॉडी में खुद का सम अमाउंट ऑफ इंसुलिन विल बी देयर व्हिच विल बी प्रोड्यूस द नेचुरल इंसुलिन, and then the person is again taking insulin from outside. So as a result of which there will be very high level of glycogen synthesis, and due to which that can lead to hepatotoxicity. यानी कि जो लिवर है उसमें एक्सेस अमाउंट ऑफ ग्लाइकोजन कभी कभी स्टोर हो जाता है, विच कॉजेस दी हेपेटोमेगली, यानी कि जो लिवर है वो एनलार्ज हो सकता है ड्यू टू कंटिन्यूअस टेकिंग ऑफ इंसुलिन. सो फॉर विच इट इज सेड दैट द इंसुलिन डोज, अगर इंसुलिन डोज अगर बहुत हाई दे रहे हैं किसी पेशेंट को फॉर सम रीजन, सोच लो कोई एक इन्फेक्शन है या कुछ भी है जिसकी वजह से हमें ग्लूकोज लेवल्स को लो रखना है, सो वी आर गिविंग अ हाई डोजेज ऑफ इंसुलिन. So at that time we have to check that whether there is hepatomegaly or not. अगर hepatomegaly हो रहा है, अगर वहां पे liver enlargement हो रहा है, then at that time the insulin should be stopped. इंसुलिन वहां पे उस टाइम के लिए स्टॉप कर देना चाहिए. The insulin should be stopped, and other than that there should be other medications also. There should be medications which will lower down the insulin which is there, not for long. क्योंकि क्या होता है ना, ये सडनली ग्राफ बढ़ने की वजह से होता है. तो अगर हम लोग एक दिन के लिए भी अगर इंसुलिन बंद कर देंगे then it will get back to normal. So that is a condition which is to be noted for the patient who are suffering from diabetes for long time. Do you understand this?
Yes, ma'am.
Yes, ma'am.
Yes.
Yes, ma'am.
Okay. Now coming to epinephrine, we have seen that glucagon or epinephrine in दोनों का काम almost same था, which means the epinephrine will also stimulate the breakdown of glycogen, but when at the time of need, that is at the time of fear and flight. So just imagine that there is a soldier or there is an athlete, right, or there is a sports person or someone who is doing vigorous exercise in a gym, right? So for them the epinephrine or the adrenaline concentrations are high. उनकी बॉडी में हमेशा ये हाई होता है. So for them what happened, that there is a continuous breakdown of glycogen. There is a continuous breakdown of glycogen, and we will see that this glycogen which is getting broken, what it is doing, it is releasing the glucose in long run, and it is releasing the glucose into the liver and to the muscles. The receptor for this hormone is present in both liver and muscle. यानी कि जब हम एक्सरसाइज करते हैं तो जो एपिनेफ्रिन सिक्रीट होता है, that epinephrine can bind to both the liver and to the muscles where the glycogen is stored, and it can act on both the organs. Fine. Now just imagine that for the athletes what happens, जो athletes होते हैं या जो trainer होते हैं ना, gym trainer जो होते हैं, so what do? They always tell the person to do a lot of exercise. तो जब बहुत ज्यादा एक्सरसाइज करते हैं या बहुत ज्यादा जब ट्रेनिंग पीरियड में होते हैं कोई भी स्पोर्ट्स पर्सन, तो उस समय उनकी बॉडी में ग्लाइकोजन ब्रेकडाउन बहुत रैंडमली होता है, राइट? ग्लाइकोजन ब्रेकडाउन उनकी बॉडी में तब बहुत रैंडमली होता है, फॉर विच देयर इज अ टाइम व्हेन देयर इज अ लीनिंग बॉडी मास, यानी कि जो उनका जो ग्लाइकोजन है वो उनकी मसल्स में से रिड्यूस होता रहता है, उनके बॉडी में रैंडम रिडक्शन होता है. सो वी मस्ट चेक कि अगर ऐसा है कि उसकी बॉडी में जो ग्लाइकोजन है वो कमता जा रहा है, देन देयर शुड बी अ हाई इनटेक ऑफ कार्बोहाइड्रेट. क्योंकि देखो ग्लाइकोजन अगर कम रहा है तो ग्लाइकोजन को कैसे बढ़ाएंगे, कैसे उसको बूस्ट करेंगे ग्लाइकोजन को? बाय गिविंग व्हाट यू कैन डू?
Ma'am, carbohydrate taking.
Carbohydrate, yes, by increasing the carbohydrate intake. So that is why mostly the one who are sprinter, जो दौड़ते हैं ना, the runners, the athletes, or even the weightlifters, it is said that the weightlifters they should have more amount of carbohydrate, क्योंकि जो carbohydrate होता है वो actually glycogen बना के देता है. अब of course if you are working, अगर तुम ज्यादा काम नहीं कर रहे हो, then your muscles will not take the glucose and convert it into glycogen, rather it will get converted into the fat. सो अगर तुम बहुत ज्यादा काम करते हो, अगर तुम ट्रेनिंग कर रहे हो, एक्सरसाइज कर रहे हो, सो इफ यू हैव टू मेंटेन द ग्लाइकोजन लेवल्स इन योर मसल्स, देन इट इज नेसेसरी दैट यू टेक द राइट अमाउंट ऑफ कार्बोहाइड्रेट. अगर उनमें हम लोग कार्बोहाइड्रेट कट डाउन कर देंगे, सो देयर विल बी अ ब्रेक डाउन ऑफ द मसल मास, राइट? सो दैट इज वन इंपॉर्टेंट थिंग that we need to remember about adrenaline. That adrenaline is good, it is helping us to get energy, but then it can also result in breakdown of the muscles.
But ma'am, what you said about it will be converted to fat?
Okay, I said that if you are not doing exercise, like say for example, you are taking a lot amount of carbohydrate, but then you are not utilizing it. क्योंकि देखो ग्लाइकोजन कन्वर्शन कब होगा, ग्लाइकोजन कब बनेगा बताओ, ग्लाइकोजन सिंथेसिस कब हो रहा था? ग्लाइकोजन
When we
सिंथेसिस,
exercise or during the stage of fasting?
नो
When the
ग्लाइकोजन,
glucose was
Sorry,
evidently
when
present
we
in the
exercise.
blood.
yes Swati,
When the glucose was evidently present in the blood,
हाँ,
that
when
Yes.
the
time the glycogen.
when the glucose was present, when the insulin was present actually, right?
Yeah, yeah.
Yes. So basically, when we do exercise, then what happens? The insulin sensitivity increases in the body, right? So for which there will be glycogen synthesis which will take place. There will be glycogen synthesis which will take place. कब? जब हम exercise कर रहे हैं, जब हम काम कर रहे हैं. But if you are not doing exercise, you are only eating and you are taking rest. So at that time, definitely your muscles are not going to build up the glycogen. But then the excess amount of carbohydrate that is there in your body, it will until ultimately give rise to the fat molecules and it will get deposited into the adipose tissue. So that is bad. But then if you're doing workout, if you're burning out your calories, if you are a sports person, you have to do a lot of work, then it is important that you get the right amount of carbohydrate, or else your glycogen present in the muscles will start degrading. Is it clear?
Ma'am, during the exercise, the glycogen breakdown occurs. That's how we get our energy.
First of all, the glycogen breakdown will start. That is from your liver, right? That is from your
Yes.
liver. हाँ, पर यहाँ
Yes.
पे हम लोग एथलीट्स के बारे में बोल रहे हैं. So for athletes, we are focusing on the muscles. So normal fasting पे हो, liver glycogen is going to break. Normal exercise कर रहे हो, then your sugar levels are getting low, so your liver is breaking it, right? But when you are losing your muscle glycogen कब हम lose करते हैं? जब हम लोग excessive काम कर रहे हैं, excessive muscle activity हो रहा है, right? So at that time, right amount of carbohydrates to be maintained.
Okay, thank you.
Is this clear to all? Is this clear?
Yes, ma'am.
Arpita,
Yes, ma'am.
Shatabdi. Okay. Yes. Now, what happened to the muscle glycogen actually? Like I said that the muscle glycogen will be broken and the blood glucose will be produced. So how it happens actually? Like you all must have heard about this Cody cycle, right? And when it happens during the muscle exercise. So when your muscles do the exercise, see, we must remember one thing. एक चीज हमें पता होना चाहिए कि जो ये फ्री ग्लूकोज का जो कंसेप्ट है ना, फ्री ग्लूकोज मतलब द नॉर्मल मोनोसैकेराइड ग्लूकोज जो बॉडी में होता है, दिस फ्री ग्लूकोज कैन ओनली बी प्रोड्यूस्ड बाय द लिवर, ओनली इसको लिवर ही बना सकते हैं, सो कोई और नहीं बना सकता, which means that in the muscle there is glycogen, but then this glycogen cannot directly give rise to the glucose. So what this glycogen can do? This glycogen can generate the lactate, which ultimately gets converted into glucose in the process of your gluconeogenesis. यानी कि जो मसल में जो लैक्टिक एसिड फॉर्मेशन होता है, एक्चुअली दिस लैक्टिक एसिड एक्चुअली गोज टू योर लिवर and then it got converted into glucose, and again this glucose comes back to the muscle so that the muscle gets energy to do the work. So that is how the cycle continues within the muscles. तो मसल में जो ग्लाइकोजन जो ब्रेक डाउन हो रहा है वो एक्चुअली कैसे हो रहा है? वो हो रहा है विद द हेल्प ऑफ ग्लाइकोलाइसिस, यानी ग्लाइकोजन किस में कन्वर्ट होता है? ग्लूकोज 6 फॉस्फेट में कन्वर्ट होता है, एंड देन ग्लूकोज 6 फॉस्फेट डज दिस प्रोसेस ऑफ ग्लाइकोलाइसिस. पर after glycolysis, what do you get? You get pyruvate. After pyruvate, what do you get? You get lactic acid, and then this lactic acid goes to your liver and it ultimately gives rise to glucose. यानी मसल्स डायरेक्ट ग्लूकोज नहीं बना रहा है, वो लैक्टिक एसिड बना के लिवर को दे रहा है, लिवर उससे ग्लूकोज जनरेट करता है. So ultimately all the free glucose that we are getting from the glycogen is generated by whom? Is generated by the liver. Is this clear?
Ma'am, can you please repeat once?
Yes, yes. Divya, anything you want to say?
Yes, yes, ma'am. When we do exercise, sometimes we get cramps or there is muscle soreness after the exercise. So, is it due to because of the lactic acid accumulation?
Of course, of course. That is the anaerobic respiration that happens, and the lactic acid is formed, and this lactic acid gives rise to that muscle cramp, right? So that is why after exercise you feel it. Correct. Yes. So here actually we have seen that how the muscle give rise to glucose. जैसे हमने क्या पढ़ा कि पहले तो जो है लिवर जो है ग्लाइकोजन ब्रेक करेगा, ग्लूकोज रिलीज करेगा, एंड उसके बाद भी अगर हम लोग एक्सरसाइज करते हैं, उसके बाद भी अगर सेल्स जो है उनको वापस ग्लूकोज चाहिए, देन द मसल विल स्टार्ट प्रोड्यूसिंग इट. बट हाउ द मसल प्रोड्यूस? मसल कैन नॉट डायरेक्टली प्रोड्यूस ग्लूकोज. तो मसल डायरेक्टली क्या प्रोड्यूस कर सकता है? लैक्टिक एसिड प्रोड्यूस कर सकता है. तो जो लैक्टिक एसिड बनता है मसल्स में, दैट गोज टू द लिवर थ्रू द ब्लड, and in the liver that lactic acid gets converted into glucose. यानी मसल्स में डायरेक्ट ग्लूकोज जो है जनरेशन नहीं होता है, लैक्टिक एसिड जनरेशन होता है, which finally gets converted into glucose in liver. See, this is clear to all or not? Chinmay, Aparna?
Yes, ma'am.
Okay.
Yes, ma'am. Clear.
Fine.
Yes, ma'am.
But ma'am, the glucose must be utilized, but it is going back again to the muscles.
हाँ, because glucose का काम क्या था? To give energy. So which part of your body needs more energy?
Okay.
Your body tissues, right? One of which is your muscle.
Okay. Okay.
Okay, yes. Now this is a important thing, and this is yeah. Anything you want to say, Shatabdi? Okay. See, this is a condition that is being shown for a person who is doing a lot more exercise. तो अगर कोई पर्सन बहुत ज्यादा एक्सरसाइज कर रहा है, which means that the glycogen levels are getting lowered, correct? Glycogen levels are getting lowered, and then as because the person is doing a lot more amount of exercise, so what happened that at that time there will be more breaking of fat and there will be less glycolysis. ग्लूकोज ही नहीं है तो glycolysis कैसे होगा, है ना? So there is less glycolysis and there is more lipolysis, more breaking of fat. But then what is the problem with this condition? It is being seen that in case of athletes, right, in case of athletes, if there is breakdown of glycogen, यानी कि glycogen continue breakdown हो ही जा रहा है, and then the person is not taking the right amount of carbohydrate or right amount of food, then at that time there will be decreased level of testosterone and decreased level of estradiol, that is your progesterone and estrogen. which means that it can even lead to hormonal disbalance. तो अगर बॉडी में ग्लाइकोजन कम हो रहा है ना, ग्लाइकोजन अगर बॉडी में कम होता जा रहा है, बट देन ऑल्सो इफ यू आर नॉट टेकिंग द राइट अमाउंट ऑफ कार्बोहाइड्रेट, देन इट कैन इवन reduce the production of the testosterone, progesterone and estrogen. So that can be very difficult for the athletes or the one who are doing vigorous exercise. So that is why right amount of carbohydrate is to be maintained. Now this is one process that we can learn during the gluconeogenesis only. We will be doing this during gluconeogenesis in the next class. Yes. Now here we will just see that what is this glycogenolysis. Glycogen synthesis we have seen. Now we will see glycogenolysis. Glycogenolysis means breaking of glucose. Now this process is just the reverse. You see what is happening here. We know that in order to break the glycogen, you need glycogen phosphorylase. तो जो ये ग्लाइकोजन फॉस्फोराइलेज होता है, व्हाट इट इज डूइंग, इट इज ब्रेकिंग द ग्लूकोज चेन. बट हाउ इट इज ब्रेकिंग, देखो यहाँ पे एक ब्रांच भी है और यहाँ पे एक लीनियर चेन भी है. तो जो ग्लाइकोजन फॉस्फोराइलेज एंजाइम होता है, इट एक्चुअली ब्रेक्स सम ऑफ द ग्लूकोज फ्रॉम द linear chain and also some of the glucose from the branch chain. यानी branch chain glucose and linear chain glucose दोनों को ही break कर सकता है glycogen phosphorylase. So you can say actually it is trimming the branch chain and the linear chain. After which it is using another enzyme, that is the debranching enzyme. Just remember what we have learned कि glycogen कैसे बना था. पहले linear chain बना था, उसके बाद वहां पे branching enzyme आया था, है ना. अब जब हम ग्लाइकोजन को जब ब्रेक डाउन कर रहे हैं, देन वी हैव टू डू द रिवर्स प्रोसेस. सो हाउ टू डू इट? फर्स्ट देयर विल बी ट्रिमिंग ऑफ द लॉन्ग चेन एंड द ब्रांच चेन, एंड देन देयर विल बी अ debranching enzyme. Debranching enzyme means the enzyme which will remove the branch from this head region and it will bring it to the linear chain. याद करो जब सिंथेसिस पढ़ा था तो क्या हुआ था, पहले लीनियर चेन बन रहा था, फिर वहां से ब्रेक होके ब्रांचिंग में जा रहा था, करेक्ट. नाउ यू सी व्हाट इज हैपनिंग, just the reverse is happening. So first the branch is getting broken and it is attaching to the linear chain, right? After which in the next step what should happen? In the next step there will be the release of free glucose. तो जो ये head region का जो free glucose था, it gets released, and after then there is activity of the phosphorylase enzyme and your glucose 1 phosphate is generated. So like we have learned कि जब ग्लाइकोजन ब्रेकडाउन होता है, so we don't directly get glucose 6 phosphate. क्या मिल रहा था हमें? ग्लूकोज 1 phosphate मिल रहा था, and then उससे हमें क्या मिल रहा था? ग्लूकोज 6 phosphate मिल रहा था. So the process which we have learned there, that is your glycogen synthesis, this is just the reverse. Just see what we have learned in glycogen synthesis कि normal glucose था, उससे ग्लूकोज 6 phosphate बन रहा था, उससे ग्लूकोज 1 phosphate कन्वर्ट हो रहा था, ग्लूकोज 1 phosphate एक लीनियर चेन बना रहा था, देन लीनियर चेन के बाद वहां पे ब्रांचिंग हो रहा था, है ना. The reverse process has occurred in case of glycogen synthesis. Now what is happening? First, the trimming of the glycogen bond is happening. Then there is the removal or debranching. तो जहाँ पे जो ये ब्रांच यूनिट है, इनको हमने लीनियर चेन पे ऐड कर दिया. उसके बाद जो फ्री ग्लूकोज था उसको हमने रिमूव करा दिया, and then slowly every glucose will be released and it will be released in the form of glucose 1 phosphate. और ग्लूकोज 1 phosphate को हमें किस में कन्वर्ट करना है? ग्लूकोज 6 phosphate में कन्वर्ट करना है, and ultimately what you get here is your glucose molecule. If you compare both the process, you will see this is just the reverse. Tell me, do you understand this?
Yes, ma'am.
Yes,
Yes,
ma'am.
Topic?
ma'am.
Yes,
Clear?
ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay, right. Yes. So just to sum up what we have learned that this breakdown will happen with the help of this glycogen phosphorylase. How it will happen? First, there will be a transfer by the group. Then there will be release of glucose 1 phosphate, and there will be ultimately release of glucose. And then what happens to the other process, that is your debranching process. In a linear chain को तो हम ऐसे तोड़ेंगे, और branching को हटाने के लिए हमें क्या चाहिए था? Debranching enzyme चाहिए था. And this debranching enzyme will make sure that the free glucose is relieved and the 1,6 linkage is broken. So in the breakdown process, there are again two enzymes which are taking part. Synthesis में दो enzyme थे, glycogen synthase and branching enzyme. अब glycogen breakdown में दो major enzymes क्या हो गए? Glycogen phosphorylase and debranching enzyme. Yes. Now, just to compare the process of this glycogen breakdown in liver and muscle. So, what we need to know, कि in liver, which enzyme is present? Glucose 6 phosphatase. And then in muscle, there is no glucose 6 phosphatase. तो हमने क्या पढ़ा कि muscle के पास glucose 6 phosphatase नहीं है. That is why what it cannot do, what the muscles cannot do, what the muscle was unable to do.
They cannot form free glucose.
Absolutely. So they cannot give rise to free glucose, which means that they have to undergo the process of glycolysis and they make lactic acid and then they give it to the liver for glucose formation. And what this liver can do? Liver has this enzyme called glucose 6 phosphatase, which it can directly use to release the free glucose in the bloodstream and to send to the other tissues. Yes. Now, this enzyme, that is your glycogen phosphorylase or glycogen synthase, we have seen both the enzymes, we have seen how do they work. So we need to remember that these three enzymes, they are definitely affected by the hormones, right? We have seen that how insulin, glucagon and epinephrine are acting on them. Other than that, there is allosteric regulation. Do you remember we have learned about the allosteric binding site for glycogen phosphorylase enzyme? Ekdam starting mein dekha tha humne that there is a site, that is allosteric site. So what happened? That to this allosteric site who will bind? Glucose will bind. देखो इसका मतलब समझते हैं. What is the enzyme? The enzyme is glycogen phosphorylase. क्या करता है ये enzyme? What it does? It converts the glycogen and it gives rise to glucose. यानी कि the glycogen will be broken and glucose will be produced. Which means that, say for example, in the blood, there is excess amount of glucose, right? In the blood, there is excess amount of glucose. So what do you think that at this point, do we need this enzyme to break glycogen and to produce glucose? Body में
No.
already glucose है, right? Yes. So body में already glucose है, यानी कि अभी हमें ये वाला enzyme नहीं चाहिए. We want to stop this enzyme. So how to stop this enzyme? If there is excess glucose in the body, it will go and it will bind to the enzyme in the allosteric site. So when excess glucose is present, it goes and binds directly to this enzyme. It blocks the enzyme at the allosteric site, for which the glycogen breakdown cannot happen. तो बॉडी में जब ग्लूकोज ज्यादा होता है, सो डायरेक्टली ग्लाइकोजन फॉस्फोराइलेज एंजाइम स्टॉप हो जाता है. सो देयर इज नो ग्लाइकोजन लाइसिस when there is high concentration of glucose in the body. Correct? Simultaneously, we will see that the glucose 6 phosphate, what it is supposed to do? It is supposed to start the process of glycogen synthesis. So अगर सोच लो that there is a well-fed condition. Well-fed condition मतलब there will be more glycolysis, there will be more insulin, so there will be more glucose 6 phosphate, है ना? जितना ज्यादा ग्लूकोज होगा उतना ज्यादा ग्लूकोज 6 phosphate बनेगा at the time of glycolysis. Now, if there is more glucose 6 phosphate, so this glucose 6 phosphate will again bind allosterically with whom? The enzyme glycogen synthase. यानी कि ये रिलेशन हम समझते हैं थोड़ा कि if there is high glucose in the blood, then the glucose will bind with glycogen synthase and your glycogen will be produced. Correct? Simultaneously, if more amount of glucose is present, then it is going to trigger it positively. If more amount of glucose is present in the blood, then it is going to bind allosterically, but to inhibit which hormone? Glycogen phosphorylase. So ultimately, your glycogen is not breaking up, so your glycogen concentrations are high. तो देखो, अगर ग्लूकोज ज्यादा है, so at that time glycogen formation भी ज्यादा होगा. कैसे ज्यादा होगा? क्योंकि ग्लूकोज is promoting glycogen synthase. तो glycogen synthase अच्छे से काम कर रहा है, there is more amount of glycogen which is getting produced, and then if there is more amount of glucose, so it is simultaneously also inhibiting the glycogen phosphorylase so that your glycogen does not break and your glycogen concentration remains high. तो जो ग्लूकोज होता है बॉडी में, इट इज एक्चुअली अफेक्टिंग both the enzyme glycogen synthase and phosphorylase allosterically, but glycogen synthase को कैसे वो रेगुलेट कर रहा है, पॉजिटिव रेगुलेशन, ग्लाइकोजन फॉस्फोराइलेज को कैसे रेगुलेट कर रहा है, वो नेगेटिव रेगुलेशन. So ultimately it is making sure that the glycogen concentration in the body, it is building up, it remains high. See, do we understand that how allosterically glucose is activating
Yes,
this
ma'am.
process?
Yes, ma'am.
Okay. Right. Okay, so here we'll just see one thing, that this is the structure of glycogen. So when you see a glycogen, you will find that it is actually a very spread out structure, and this spread out structure is because of the branching. So this branching is actually giving rise to such a spreading out structure. Now, this branching pattern, it actually increases the solubility of glycogen, which means that more amount of glycogen can be trapped in the muscle. लाइक व्हाट इज ग्लाइकोजन, ग्लाइकोजन इज अ पॉलीमर. क्यों बनाते हैं हम पॉलीमर, ताकि हम बहुत सारा ग्लूकोज जो है एक जगह पे हम लोग स्टोर कर सकें. अब ये जो ग्लूकोज हमारे पास है, वी आर स्टोरिंग इट इन द फॉर्म ऑफ ग्लाइकोजन, करेक्ट. बट इफ द ग्लाइकोजन इज अ लीनियर चेन, अगर सोच लो कि ये ब्रांच नहीं होके अगर लीनियर होता, देन द मसल्स वेर अनेबल टू एकोमोडेट इट विद इन देमसेल्व्स. सोच लो तुम्हारे पास बहुत सारे छोटे छोटे पैकेट्स हैं, and what you're doing is you're arranging them in a linear chain. So what will happen, your space will be limited, you cannot keep more amount of packets. But then if you are making stacking, if you are stacking them one after the other, then you can keep a many more number of packets in the same space. In the same way, the glycogen, it is creating branch so that more amount of glucose can remain in the muscle, and that is how it increases the solubility of this glycogen molecule in the muscle cells or in the liver cells. So basically the branching is important so that the solubility can be increased. Yes. And we have
Ma'am,
again
how
seen,
is branching related to solubility?
it actually relates to the surface area. It actually relates to the surface area. Achha, tell me one thing, in the polysaccharide class, have I told you the function of glycogen there? Just remember and tell me, have I told you about the glycogen, that why glycogen, osmotic regulation I've told about glycogen? Anyone remembers? Osmotic regulation. I
I
generally
don't
tell this.
remember, ma'am.
Fine. Okay, I'm just telling this to you in case I have not mentioned it there. Just remember one thing, that there is an osmotic pressure which builds up in the muscle cells. हाँ, क्योंकि देखो हम लोग क्या कर रहे हैं कि हमें क्या पता है that every cell or every biological unit, it has a osmolarity
Ma'am,
or
excuse
हाँ,
me, ma'am. Excuse
yes,
me, ma'am. Well,
yes,
little bit you have said that the glycemic is stored in the polymeric or condensed form to
correct.
minimize the osmotic effects like that.
Right, absolutely. I'm talking about that only, right? Then I have mentioned that. Just please go and check it once in the video. Yes. So just we need to remember one thing, that this glycogen, which is a polymeric form, it has to be branched. क्योंकि अगर branch नहीं होगा, so ultimately the proteins which are there in the muscle plasma, they are not going to hold the long chain. तो अगर इस long chain को हम रखेंगे muscle के अंदर, तो यहाँ पे suspended नहीं रह पाएगा, right? So what will happen? It will not remain suspended, it will precipitate. So we have to make sure that the structure of glycogen should be such so that it can remain suspended in the muscle cells or in the liver cells, for which it take up the shape, that shape which I've shown you, and that shape is possible because of this branch structure. So if there is loss of branching in glycogen, then there will be opening up pattern of the glycogen chain, for which the glycogen will get precipitated in the cells. It will not remain suspended. Do we understand this? The structure of the glycogen is made by this branching pattern. तो अगर branching pattern हटा देते हैं तो वो structure नहीं रहेगा, structure नहीं रहेगा तो वो उन muscle proteins के बीच में उस surface medium पे वो वहां पे suspended नहीं रहेगा, वो precipitate हो जाएगा.
Ma'am, what you were saying about the osmotic pressure?
Osmotic pressure, I've mentioned in that class that glycogen plays a vital role in order to maintain the osmotic pressure of the muscle cells. And I've mentioned that the relation with N, you remember that if there are more number of molecules, so the osmotic pressure is
Yes,
more.
yes, yes,
Yes,
yes,
exactly.
yes,
And
yes. That I
directly
remember.
proportional to
Correct,
like that.
correct, correct. Yes. Now coming to the hormonal regulation, though we have discussed is one, just see that in this case, glucagon and epinephrine, what they will do, they will activate and insulin will inhibit, just the reverse of glycogen synthesis, जो हमने देखा था कि अगर glycogen breakdown है तो insulin inhibit करेगा, glucagon और epinephrine they are going to promote it. Okay, yes. So that was all about your glycogen synthesis and glycogen breakdown. Do you understand this?
Yes, ma'am.
Okay.
Yes, ma'am.
Any query from today's session, whatever we have discussed till now?
No, ma'am.
Okay, fine. Right. I will be sharing the link because I have experienced that many of you are having problem because you don't get to revise it. That is why. So fine, I will be sharing the link. Just do check it once and also prepare for the next test. We will have another test from this part, that is your glycogen and glycolysis. The previous class and today's class. We will have a test in the coming week. I will inform you when we will have it.
Okay,
Okay,
ma'am.
ma'am.
Okay, ma'am.
Okay. Okay.
Thank you so much.
Thank
Thank
you.
you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you.